Welcome, everybody. I think we probably should get started because I know Dr. Patay has some really incredible stuff to present today, so I won't take much time. I just want to take the time. My name is Jeff Miller. I'm the CEO and Chief Scientific Officer of Invivoscribe, and I'd like to introduce Dr. Brad Patay, who's our Chief Medical Officer. And I can say that he's board certified in internal medicine, pediatrics, and he's an active clinician as well.
Good morning, everybody. I thought I'd start off this slide with a recent issue of Nature. But, a lot of people read Nature, right? Policymakers, scientists, maybe even people with cancer. And so I thought there might be a little rosy bias because Illumina was sponsoring basically the content. So I naturally am interested in oncology, so I flipped to that slide. And I started scratching my head. I was like, the precision oncology illusion. Precision oncology has been shown not to work, and perhaps it never will. Wow. Precision oncology is inspirational. What doctor or patient would not want to harness genetics to tailor a therapy to an individual? But traveling back in time machine is also inspirational. Who would not want to wind back the clock to remove the cancer before it spreads? In both cases, however, as of 2016, the proposal is neither feasible, cost-effective, nor assured of future success. Yet, in only one of these cases, does the rhetoric so far outpace the reality that we risk fooling even ourselves. Wow, ouch. So, that struck me as, pretty strong comments from a hematologist oncologist at Oregon Knight, who's practicing physician. So why do you say that? You may know the Shiva trial is an academic trial of 745 patients of solid tumors. They're a heavily pretreated group. They used basically molecular assays, targeted therapy, and showed no difference in precision in basically event-free survival. There was also an NCI match trial that he cited that essentially had a 6.5% success rate. But, let's maybe look at some things because, one, we really need to figure this out. There's about 45,000 people that die of basically non-Hodgkin's lymphoma, leukemia every year. And I think the chemotherapy, we're really hitting the wall against that, and we really need to look at targeted therapy and immunotherapy as treatments. As we know, precision oncology definitely works, right, in some cancers. And as we know, when you look at essentially CML, it skyrocketed upward the overall survival. And that was due to imatinib Gleevec, right? A targeted therapy. We need to offer hope, I think, because if you look at acute myeloid leukemia, the rate is essentially 25%. And so if we are capped out at 7+3 effectiveness, and we're really looking at better therapeutics, I think we're going to need genetics to help us. There's a lot of drugs in development right now. And as we know, if you don't design your trial right, have good randomization of the cohorts, then you could really have a miss on your drug and submission. Even though there's about over 200 drugs in hemopathology, hardly any make it through approval process. So I think to say precision oncology is illusion, no, I think it's definitely a reality. And what I hope to do today is to show you that I think we're going to need, in clinical trials, definite more genomics or understand the genetic architecture. And what we really have done, and now in 2016 going to 2017, we really understand much of the genomics, basically AML and many of the other leukemias and lymphomas. One more, I just want to keep hammering on targeted therapy for a second because it's not an illusion. I don't think illusion of saving patients. This is the APML3 trial. APML is basically a cohort from Australia and New Zealand. And essentially, they're using targeted therapy with ATRA and ATO to improve survival in APL patients. This trial was novel in that in low dose, it had been shown that ATRA, ATO improve overall survival compared to chemotherapy. And what they did is they lowered the dose of anthracycline in the highest risk groups and improved the overall survival. So yes, definitely target therapy works. I think if you talk to an APL patient, they would agree with that. And I just want to highlight this slide in the power of clinical trials. I think if you do sort of take a hodgepodge of patients and you only look at a few key therapeutic pathways that really don't have good therapeutics, yeah, you might not have a good event-free survival or overall survival. But I think in well-done clinical trials, with a full understanding of the genomics, I think there's a much better chance. You know, I'm lucky to work in Invivoscribe. We work with many of the key thought leaders in AML, and I'm particularly proud of this one trial headed up by Pam Becker and University of Washington, Seattle Care Alliance, where essentially she's using our MyAML assay to basically show that it's both feasible. This is sort of the overall primary endpoint, that within 21 days there can be a chemosensitivity assay, a mutation panel on basically relapsed refractory AML patients, and using a whole host of potential different target therapy, can that be delivered to the patient in 21 days? And I'm looking forward to see that data, but we've been working with her for quite some time. The full, this trial should end in 2018 to 2019, but we've been doing it for well over a year now. So again, targeted therapy, we all know that FLT3 is a big story last year with the Ratify trial. This is Richard Stone showing that at ASH 2015, showing a 23% basically survival benefit, overall survival with mitostatin, and this FLT3 inhibitor should be being approved soon by the FDA. Again, there's other FLT3 inhibitors that are coming out and going through both induction consolidation chemotherapy regimens with the 7+3 and also relapse refractory. So I think if you hear people give like, well, there's 18,500 maybe patients had targeted therapy done, you want to really have the right assay for the right cohort and the right treatment sort of armamentarium before you say that essentially genomics and precision oncology is illusion. So AML lags behind everybody else. But as we know, I think the key with gene panels is understanding that not, even though your morphology this maybe suggests you're within this certain phenotype, that's like, I think, a little bit of old thinking. Because if you think about it, you know, AML, depending on if you have APL, you would be really high overall survival. If you're a core binding factor AML, you may be in this range. And then unfortunately, if you have a complex karyotype, TP53, you're going to be really poor. So I think what, as pathologists in the heme space, we really need to push essentially what type of AML. And as we know, essentially, you know, there's a lot of agreement on how to treat poor risk, potentially young people, take them to transplant. And also, I want to just note that for precision oncology being illusion, we use now, this is a new addition to the NCCN guidelines about TP53. You're looking at FLT3 molecular testing, biallelic CBP alpha, NPM1. So that bone marrow, you have a little slice of that. Right now you're all cutting it up, sending it to different assays. Maybe it's time, almost the tipping point now in terms of cost and sample specimen to start thinking about gene panels. And AML is one of the sort of easiest ones. So that's why I'm focused on a lot of it today is if in nature they say it's illusion, let's talk about some of the easier ones before you say solid tumors. I don't think solid tumors may be the best example of target illusion, although there is quite a bit of target therapy for lung adenocarcinoma. So I think this slide's critical because if you're designing a trial and you think you have good randomization of the patients, you match them by age cohort, basically Karnofsky performance scale and others, but if you don't have essentially like a TP53 testing, those patients essentially don't make it past a few years. It's a very poor prognosis. So you've got to have that testing. And then I think it's really interesting, and I'm going to touch on this in another slide, is look at RUNX1 mutations. And this isn't the fusion protein, RUNX1, RUNX1T1. This is just RUNX mutation, how poor that overall survival is compared to what we know as core binding factor and NPM1, some of the better, and biallelic CBP alpha. And then here's essentially just in May this year how RUNX1 is essentially a new provisional entity. So I think these slides are take-home point is genetic testing isn't slowing down in genomics. It's accelerating. And when's the tipping point that, gosh, maybe it's not all about single tests. Maybe we should be moving to panels. Again, here shows this whole scope. And it's not even just detection of the variant. It's the clonal architecture, right? How are the genes interacting? What's the dominant clone? And understanding the gene variant in the context of the clone itself, I think, is almost more important. But I really wanted to touch on this. I think, this is the era of big data. And what is big data? It's really modeling, machine learning. And in clinical medicine, if you're below 0.7, that's not a good model. It's not helping predict risk. And so what I see is when you are taking full models, we're moving it into the fair. If we are 0.8, 0.9, those are great models. But I think what you're seeing is if you're taking clinical variables, demographics, and then these gene panels and maybe some other proteomics work in the future, then we'll be really able to help predict and meet the heterogeneous AML to the actual targeted therapy that we want. This again just shows you that, for example, FLT3 ITD is significant in the model and has a large effect size. Maybe not as much as TP3 inverse 3, but essentially there's mutations that need to be picked up and tested for. Okay, so I'm going to just move into some analytical work we've done with our gene panels, both our MyAML and the MyHeme pathway panel. So MyHeme, essentially we created it. The trick with panels, right, is you can't have a panel that does everything. You got to really pick up panels for specific reasons. And the MyHeme essentially looks at all of hemopathology, lymphomas, non-Hodgkin's, Hodgkin's, essentially looks at all the leukemias, multiple myeloma. And we have 571 genes that we go over the coding region. And then we use RNA to pick up essentially all the fusions that have been described in hemopathology, picking up the structural variants. And then we add clonality as an option to the MyHeme panel. And essentially, we have very good coverage. We can pick up the 2.5% variant allele frequency, which has been shown in studies to be significant for implications for minimal residual disease and further risk classification of the patients themselves. Here's MyAML, which we've done many samples. Again, this one's slightly different in that we test the DNA, not the RNA for the fusion, so we can actually give you the actual breakpoint location, not just over the intron itself. And like both, we have excellent sensitivity specificity, which I'll show soon. So, but it's much more than that. Like you can have the assay, and then the bioinformatics is really key. And then even maybe more for pathologists is the communication. And we offer this as a service because, and we're not kidding it, because we really want to be dialoguing with the ordering pathologists and the ordering oncologists. I think there's a lot of education that needs to happen. And I think all of you being here in this room is exciting because I can't tell you how many variants that we pick up that aren't classic drivers. There's this one really rare mutation, it's called a point mutation in the junctional membrane, V592A, happens about 2-3% of AML patients. And we picked up those twice. And that may be a variant that people aren't aware of. But in studies in the labs that demonstrated that, midostaurin actually worked against that variant. And that's something that's not going to be picked up in treatment. So we want to really communicate and discuss with physicians the different pathogenic variants and the pathologists. So best-in-class bioinformatics, we really spent a tremendous amount of time in bioinformatics. Coverage is critical, particularly of the areas you're most concerned about. Essentially, here's MyAML, MyHeme. You're going to need at least an average 1,000 coverage. But you also, and what we've really done is there's certain areas of the genome that are difficult to sequence, right? Because of GC content, for example, CEP alpha. And if you read a study and you see, oh, this panel covers this disease, really get into, like, well, how much coverage was actually, like, essentially over CBP alpha? Unfortunately, GC content tends to be really biologically active. It really drives often disease. So if you're low in that, you're often missing many of the drivers. So we spend a lot of time. You have to optimize chemistry. You have to really look into the haplotypes of the gene itself. So it's much more than just detection. It's like optimizing every step of the sequencing platform for the variant you're going after. Again, here's MyHeme. We're really excited about essentially the coverage that we're getting at the 2.5 percentile. Basically, this is the entire non-coding and coding. So sometimes the non-coding phase just a little bit. The percent targeted coverage over the exons, you're at 98%, which we're really glad to see that coverage. Sensitivity specificity is excellent in MyAML. I think we've really demonstrated that when we also compare it to the pathologic reports and the communication between our customers and the assay. Here's the MyHeme sensitivity specificity. Again, at 2.5%, essentially have excellent sensitivity, specificity, and the indels at 5%. Excellent. This is the assay compared to genome in a bottle. The people at NIST that talk about genome in a bottle, oftentimes the specificity is a little bit lower because what happens is you might have a SNP or an indel really close to another one, and it can throw off the calling, particularly at very low variant allele frequencies. So there's certain pathogenic variants that you have to really even consider the surrounding area. And we have steps with our annotation team that we really look to make sure that we're not getting spurious calls. So there's a lot to NGS assays that you have to spend a lot of time and really know every gene and what can happen and even the locus within that gene. Another thing I really want to highlight is not every assay is equal, and not only in terms of depth, but also in terms of the read length, right? So you read a certain out, and then if you're only reading a little bit, it's really difficult to pick up insertion deletion sequences. So for example, FLT3, essentially right in this one area, there's a large insertion deletions, and they can go up to essentially 1810 or even 300 base pairs. So you have to have really good long read lengths to pick up these long insertion deletions. And we see this in COSMIC. So COSMIC is the catalog of online somatic mutations in cancer. And I think there's an underrepresentation, underdetection of FLT3 ITDs in the literature because they often use smaller read lengths of maybe 100 base pairs, 125. You really need long read lengths to pick up FLT3 ITDs. So go to the methods or talk to your person about the read length, like what is the detection rate. And here for us, sensitivity specificity, essentially we have excellent positive and negative controls with this locus. That is critically important with AML. Going on to MyHeme, so again, detection of fusions, translocations, we're looking at the RNA. Not the DNA, and we essentially demonstrate in different cell lines. This is a RUO product right now. It's not CLIA-CAP. We love to, if you're interested in research with this assay, please contact us or go to the poster for more questions. But we can essentially pick up many critical drivers of leukemia, and we use various different calling algorithms to the detection of those variants. So just to highlight that, for example, KMT2A, in the MyAML assay, we go over all the major hotspots, and we sequence over that. So we can actually give you the exact breakpoint, which has implications for further potential follow-up MRD assays that you could use. And I think it's critical for the community to pick up all translocations that essentially drive the biology. For example, NUP98 NSD1, it's a very difficult translocation to pick up. It's missed by cytogenetics because essentially you're fusing the terminal, this end of the P arm with the Q, and so you're getting a really small, essentially, translocation. that's often classically missed, which can easily be picked up by sequencing assays. Again, here's essentially a slide that we've had for a while showing a lot of critical development work we did, picking up all major translocations in leukemia. And really, again, cytogenetic has lots of limitations. It can't pick up cryptic gene fusions. And then, as we may know, PML rara, which essentially can be cured in almost all people, but about 5% have these cryptic complex structural variants that this assay can pick up. And again, at least there's 5 different genes that recombine with KMT2A or MLL in the 19p, 13.1, 13.3 region that are difficult to clarify with classic cytogenetics. So again, calling CNVs, this is active research in terms of it's not a clear cap as part of the MyAML, but we're moving towards that where we can, we're acquiring basically the samples and showing the sensitivity, specificity, and picking up these type of variants for the driving of essentially leukemia, and we'll have that with the MyHeme panel too. Here's an example. We have been working with a company, Epizyme with EPZ5686. That's a .1L inhibitor. And I think it's really, you know, your therapeutic agent, I think it's, if you're going to spend that much time, money, effort into developing the drug, you really want to understand and really fine detail what's happening with each patient. And for example, this is critical for to understand what's happening with the .1 interaction with the fusion protein because the different fusion partners interact with this enzyme differently, and this drug has different efficacy. So for example, they gave us a patient that was just a T311. They thought that probably would work. It's a KMT2A translocation. But we actually show that it was non-classic because it doesn't contain the 5' KMT2A part of the gene, so it's not a regular LL fusion. The fusion consisted of a TFG with KMT2A on the 3' end, which would basically makes that the Epizyme drug wouldn't work. And so this patient probably shouldn't have been enrolled in their studies. So the assays essentially, you know, you have to spend a lot of time in every one of these to optimize to make it feasible. You got to make it fast, right? We have to be in clinical time. So essentially, we can do this in about 7 days. We give 14 days, 7 to 14-day window, because if the assay fails or if the sample isn't good, we'll retry. But we're trying to get this out in 7 days, so it impacts clinical care. So I think that's important, is how fast is it? So turnaround 7 to 14 days. And the specimen is not that large, about 700 nanograms or 500, depending on which assay we're talking about. And that's really within clinical medicine. So it's not only feasible, and it's cost effective, I think. When you're talking about $200,000 for a bone marrow transplant at 100 days, I think, and all the assays you already have to cover, I think we're getting to the point where maybe we should be talking about assays in every patient for particular leukemias, particularly heterogeneous, poor outcome phenotypes. So, questions?